Good morning. Rotterdam is home to beautiful and famous bridges. In our research group, we also build holistic bridges between patient care, diagnostics, and research within our department, nationally and internationally. In the next 15 minutes, me and 3 other young principal investigators will tell the story of our department on behalf of the group. We will illustrate the research we work on, our recent achievements, and our shared view of the future for the department. I will start off with an introduction of our research and our patient population. Chaco will tell you all about our experimental wet lab research and its applications. Hiab will talk about the next frontiers in our fields, the use of big data and studies on a non-coding genome. And Sam will explain about our work aiming at educating and communicating our work to society. My name is Virginie Verhoeven and I'm a clinical geneticist. I lead one of the research group on heritable eye diseases at our department. I'm one of the 5 awardees of a VENI grant, the most competitive national early independent investigator grant. And I'm one of the 5 awardees of an Erasmus MC fellowship, a renowned grant from our own institution. In our outpatient clinic at our department, we see patients and their family members. They ask me questions like, what disease do I have? Is it heritable? What is the cause? And is there a cure? And what is the risk for my offspring and family members? And is there anything I can do to make sure I don't pass it on? Our patient population and research group is very diverse in many ways. First, in terms of disease type, varying from rare diseases, so prenatal genetics and congenital anomalies, cardiovascular genetics, neurogenetics, and metabolic disorders, as well as cancer. We are closely connected to Sophia's Children's Hospital, the largest children's hospital in our country, which gives us access to a unique set of patients. We work closely together with other departments, for instance, in various multi-expertise groups involving clinical geneticists, laboratory specialists, other medical specialists, and researchers. Second, we are diverse in terms of age. Patients vary from prenatal to children to very old. And third, in terms of ethnicity. Rotterdam is a melting pot. Approximately 50% of our Rotterdam patients have an immigrant background. And as you can see, our group of PIs is also a great mix of people with different ages and background working on various diseases. Genetic diseases can have different genetic backgrounds, ranging from monogenic to polygenic. Monogenic diseases are caused by a single genetic variant with a large effect and high penetrance, and polygenic diseases are caused by an interplay between many genetic variants. An important focus of our department has traditionally been on rare diseases, but we also focus on common diseases, such as dementia, myopia, and cancer. Most of our patients have diseases with an unknown cause. We do everything we can to make a diagnosis. Usually, individual patients form the basis of our research questions, and when routine diagnostics fail, they turn into a research project. Now over to Chaco.
Good morning, my name is Chaco van Ham. I'm a research group leader, and as Virginie Verhoeven told you, we have a very large, very diverse patient population, and once we identify some of those patients that have novel diseases or diseases for which there's no good treatment, then we further investigate them in depth using the different models that I will explain you about now. So in my research, we mostly use zebrafish as a model to study different glia cells in various brain diseases. But in addition, I'm also heading the functional genetic diagnostics unit, where we are trying to bridge the techniques that we develop in the research and try to apply them to benefit patients. And so what typically happens in our research department is that we have patients that have a new type of mutation or a gene that's not previously linked to a disease, and then we investigate that further. So for example, we can look functionally if the gene is expressed in that patient in the right tissue, or we can look at the variants affecting protein function, and this helps us to diagnose this particular patient, and this also has impact on the way those patients are treated, and this also helps future diagnoses of patients that might have similar variants or similar genetic mutations. For example, the research of Maria Alves, who's shown here on the slide, has used this strategy to identify several new genes that, when mutated, cause congenital intestinal disorders. And in this research, they have access to unique surgical patient tissues that they use for this. And I think it's a very nice synergy between the clinic and the surgery department and the molecular work to help benefit those patients. Once we have a diagnosis of the patient, we want to look further into the mechanism, and we can use this, for example, using zebrafish. So what we typically do is we use CRISPR-Cas9 to mutate the corresponding homologous gene in the fish, and then we look for disease-related phenotypes, and those can be more macroscopic phenotypes or cellular phenotypes that mimic the disease in the patient. And this allows us to investigate really in depth the mechanism of the disease at the cellular level in the patient. An example you can see here in this movie where we discovered a new type of leukodystrophy caused by mutations in CSF1R. And if this gene is mutated, then you can see in the fish that the green cells, those are the macrophages that migrate into the brain to become a type of brain cell, and they fail to do this in the CSF1R mutant fish. And so this is basically the mechanism of this disease, is that the microglia are lacking in those patients. And the impact of this disease it was published in a scientific journal, but it's also in a public science journal called The Atlantic. The boy missing an entire type of brain cell. And the clinical impact of this is that this type of insight helps identify treatment for diseases like this, where now we know that those green cells, the macrophages, are basically blood cells, and they are lacking. And now we know that the hematopoietic stem cells can be used to treat patients with these type of leukodystrophies. In addition, those cells are very important in Alzheimer's disease, so this insight is also very relevant for that. In the last few years, we've made about 10 new genetic models in our department for various diseases. So when we have those models, we typically, for example, an iPS model, induced pluripotent stem cell model, we want to use them to develop therapies to better treat those patients. So for example, the group of pin pineapples shown in this image here uses iPS cells derived from patients to make new muscle cells on a chip, and they can actually contract in a dish. So they're made from skin cells and they're real muscle tissue. And he uses this to study a disease called Pompe disease, an infantile, very severe muscle disorder, to generate new therapies and based on RNA and gene therapy. And he's filed several patents in doing so. So having shown this, I guess the future in the coming years, we will focus more on making even more specific models in zebrafish for our patient. We call them zebrafish avatars, and to use iPS and zebrafish models to develop better therapeutics and leads for therapeutic treatment, and to better continue to integrate the clinic, the diagnostic, and the research work to benefit patients, as we have done in the last few years and continue to do. And now I give the floor to Hiab, who will talk to you about the next frontiers that we encounter in the research we do.
Hi, I'm Hiab Adams. I'm a clinician and genetic epidemiologist. I lead a group that studies common diseases of the brain and cancer. Chuck just showed the extent to which our researchers go to help patients with monogenic disorders. However, as you can see in these graphs, monogenic forms cause only a fraction of disease for many common disorders, whereas the majority is polygenic, so caused by the interplay of many genetic variants. This is true for brain diseases such as Alzheimer's disease, various forms of cancer, as well as many other disorders that we study in our department. And if we want to help those patients with a polygenic disease, we need to understand what the genetic causes are. Our department has had leading roles in the identification of genetic variants contributing to common diseases. The teams of Virginie and myself have led numerous international efforts on eye diseases and brain diseases, and also actively contribute to studies of heart disease, lung disease, and traits such as height and weight. This leading role is also clear by the fact that many of our collaborators send their samples and data to Rotterdam, and our department has become a central hub for genetic data analyses. Specifically, We have a strong bioinformatics core that has developed novel statistical approaches and software for the analysis of genetic data. And we were generously supported by NWO with a major infrastructural grant to do big data analysis for polygenic diseases. The genetic discoveries for these polygenic disorders have made one thing abundantly clear: the importance of the non-coding genome, the 98% of the genome that does not code for a protein. While it was previously known as junk DNA, we now know this part of our genome is actually quite important. However, the function of the genetic variants in this non-coding genome remains obscure, and the lab of Stefan is dedicated to understand what goes on in this poorly understood part of our genome, and he's using state-of-the-art methods to determine the role of these genetic regions with a particular focus on the brain. We also want to go beyond understanding, to using this knowledge to help individuals, including to determine who is at higher risk of a disease and which treatment would have the best effect. And our department is actively leading efforts for clinical implementation of polygenic risk scores within our hospital. This will bring us closer to personalized medicine, since every individual has a unique genetic background. And as Virginie mentioned, we are also unique because of our ethnicity. Unfortunately, the unequal representation of ethnicities in genetic studies has made the benefits of this advance less for most non-European groups, and we feel strong responsibility for tackling this issue, least of all because we work in Rotterdam. Among other things, I initiated the United Consortium in 2018, which focuses on the dementia research in underrepresented populations. It has already grown to the largest collaborations of its kind. With efforts like these, we want to ensure the impact of our work is felt in all layers of society. And Sam Riedijk, the final PI to present, will tell you more how we translate our genetics research back to society.
Hi, my name is Sam Riedijk and I'm a medical psychologist and assistant professor working in clinical genetics. In the next few slides, I will show you why and how we're using social science research to bridge the gap between genomics and society. As this cartoon illustrates, this is how many of us are used to deal with issues in genomics. However, not only our ideas, also those of our patients are extremely important, and even the public may have questions and desires we should take into account. That is why our adagio is let's start talking with people instead of about them. And I will show you how we're applying this in our research. In our prenatal genetics, we try to identify the genetic cause of fetal anomalies on ultrasound. If you would compare the detection of genetic aberrations to a fishing net, we have moved from wide maze nets, catching big aberrations, to very fine maze nets, catching aberrations at base pair level. Of many of these aberrations, the meaning is currently uncertain. And we studied the impact of offering a broader scope of genetic testing by offering pregnant couples real-life choices, as opposed to hypothetical choices between narrow or broader scope prenatal testing. And this is quite a unique approach. In addition, we conduct research with pregnant couples, medical doctors, and laboratory specialists to determine how to deal with uncertainties associated with exome sequencing in prenatal genetics that are impacting each of them. And as you can see here, we do this within an international consortium. Besides that, a new technology has entered the field of genetics, CRISPR-Cas9. This technology to cut and replace genes was discovered by Jennifer Doudna and Emmanuelle Charpentier in 2012. And just last week, these researchers have been awarded with the Nobel Prize. And I was invited at national radio to reflect on this, as you can see in the picture. CRISPR is used by researchers around the world, and promising clinical trials for the treatment of hereditary diseases like sickle cell and hereditary blindness are emerging. CRISPR can also be used to alter the DNA of embryos. This means that adjusted DNA can even be passed on to offspring. Gene editing thus has the potential to impact our species, which raises many ethical concerns, and therefore public dialogue about what to do or not to do with this technology is much needed. That is why we and 5 other institutes have started a national approach to study how the public may best be engaged in dialogue and to form an opinion. And it's the synergy of our genetic expertise and our clinical expertise that enables us to drive this societal dialogue. Here you can see what a dialogue looks like, employing an informative format and reaching a broad diversity of subgroups, even children here in the Sophia Hospital. Bridging the gap between genomics and society is a new research domain that requires synergy of social science and biomedical sciences research. In order to further this new research domain, our excellent research groups have taken the responsibility to develop the 2-year interdisciplinary research master, Genomics and Society. From the presentations of the 4 of us, I am sure that you will recognize that we are a curious, proud, and ambitious group of PIs, and with each other's unique expertise, we plan to integrate the research in coming years even more to meet the needs of our individual patients, families, and society.